Okay, so I'm going to do a quick overview about muscles. So we have three types. We have cardiac muscle, skeletal, and smooth muscle. So cardiac muscle is, around the heart, and it's composed of 1 to 2 nuclei. It is involuntary and it is striated. Okay, then we have skeletal muscle, which is, unlike the other two, voluntary. That means we can control it. It has many more nuclei. It is striated, and it consists of sarcomeres, which I'm going to go over in a little bit. And then smooth muscle. Smooth muscle usually lines the blood vessels, and it consists of 1 nuclei and is involuntary and non-striated, hence the name smooth. Okay, so let's look at what a sarcomere is. So a sarcomere is a muscle unit. It consists of Z lines. Z is the letter that's at the end of the alphabet. Therefore, it flanks the ends of the sarcomere. One here, one there. Then we also have actin and myosin. So actin is a thin filament, and it's denoted by the letter I. Okay, so I for thin, it's a thin letter. And then we have myosin. Myosin is a thick filament, and H corresponds to myosin in this diagram. I'm going to explain now. Think of H as a thick letter as opposed to I, which is thin. So myosin H, actin I. So let's look here. So we have the I band. So this is actin. All of these lines are actin, and the ones in the middle, that's myosin. So H consists of all of the only myosin area that's not overlapping. Okay, so we're not talking about this area that's overlapping. We're talking about this area that's not overlapping, and this is myosin. And then the thin filaments, okay, is recorded by the I. The I is talking about all of the actin filaments that are not overlapping. So for example, this area would be one example. And then we have the A. We can think of the A as in all of the myosin or the thick filament. And as I explained, the myosin is usually this one. So A for all of the myosin, it's going to be from here to here. Okay, so what usually happens? So the brain from the motor cortex is going to send an upper motor neuron, and then that's going to synapse with a lower motor neuron, and the lower motor neuron is going to release acetylcholine. Okay, and then acetylcholine is going to release into what's called the neuromuscular junction. As it implies, it's the junction between a muscle and a neuron. So acetylcholine is going to get released, right? And this is done through calcium-gated channels, that they open, and then that allow the acetylcholine to be released into the presynaptic cleft and be uptaken and induce a conformational change in the sodium channels that allows depolarization of the cell. And then we get an action potential that's going to travel from the sarcolemma. The sarcolemma is like a membrane surrounding all of these muscles, and eventually to the T-tubules. The T-tubules are just an extension where they travel down. So it's transferring that action potential, and eventually it's going to reach what is called the sarcoplasmic reticulum. So think of the sarcoplasmic reticulum similar to the ER, and in storing calcium inside of it, and when it senses the depolarization, calcium is going to be released from the sarcoplasmic reticulum out, and then we get this reaction. So as calcium concentration has increased on the inside, calcium is going to bind to a molecule called toponin. Okay, I usually remember calcium binding to toponin because topo is like on top, and then in as in it wants calcium in. So calcium is going to bind to toponin, and toponin is on top of topomyosin. Okay, so topomyosin, why is it called topomyosin? Because topomyosin is kind of covering the myosin binding site, and this is all on top of actin, so they're stacked against each other. We have an actin filament, which is a thin filament with toponin that binds calcium, and then topomyosin is going to do a conformational change. It's going to release the myosin binding site. Okay, so we need a calcium to release this myosin binding site. Next, we have myosin. So myosin is now going to be able to bind, but wait a minute, the only way it can bind after hydrolysis of ATP though, because we need ADP and phosphate to be bound to myosin so that it can bind to the myosin binding site. Next, we get the release of ADP and phosphate, so everything falls off. It's like pew, falls off, and then we have poor little myosin alone, so it's going to contract. Okay, this is muscle contraction right here. It's actually the power stroke. The power stroke is dependent upon the release of ADP and phosphate. Okay, so that's going to go down. So now we get contraction. So it's pulling. What this is doing is myosin is attaching to these actin molecules and pulling it in a way so that it can contract and shorten these sarcomeres. These sarcomeres that you see over here, we have myosin in the middle, then we have actin here. So myosin with calcium and ADP and phosphate is going to bind, it's going to attach to that thin filament right there, the actin. It's going to pull the whole thing towards itself. And that's what causes muscle contraction. Anyways, moving on. So the next step is that ATP is going to bind, okay? When ATP binds, this little guy is going to release actin. So myosin lets go of actin. It says, I don't want you anymore because I have my ATP bound. So when ATP bound, it just like falls off, it slips off. And then eventually we get ATP hydrolysis. So we go from ATP into ADP and phosphate, which when bound to myosin and in the presence of calcium induces another contraction. And there you have it.